within the growing season. And if we have fewer generations of nematodes, maybe that means we're going to have a better yield. The big question is, how do you differentiate life stages? Let's hold our cell phone calls to the end, please. Just joking. All right, so one of the ways that you look at nematode development is you stain the roots. And then you look at the nematodes within the roots and try to make this judgment call of what life stage is it. That's really hard. The way you're going to tell nematode life stage, count cuticle layers, look at gonads. You're not going to see this in there. All right, so if you macerate it out and you pull the nematodes out, and then you're looking at it there, you're going to have a ton of nematodes to look at through a ton of root debris, and that's still not going to be very effective. So I came across this method. What if we use sieves? Sieves objectively isolate different developmental stages based off of nematode size. Like us, as nematodes get older, they get bigger and fatter, so we can separate them out. So we can isolate out second stage juveniles using, say, a number 500 sieve, J3s using a number 200 and 325, J4s using a number 100, and adult females using a number 60 sieve. We can grow these seed treatments in naturally infested soil and look at the development of it after 10 days of growing, say 20 days of growing, and 30 days of growing. Macerate the roots, pour that slurry into a sieve stack, flush water through it, and then count the number of nematodes that have been isolated on each one of those sieves. And then we can look at the distribution of those developmental stages across treatments and time. Males, there's a little bit of a challenge with this. So coming back to our earlier SCN background, which I told you so well about, males leave the roots to search for females to mate with. This method will only collect males that are still within the roots. So males that have left the roots, unfortunately, can't be counted in this. So we're not going to look at the males for this development data. Again, I'm going to look at proportions. So the percentage of nematodes for a given treatment on a given sieve compared to that of untreated Williams 82 on that same sieve. And again, more than one means our treatment didn't work. Less than one means our treatment worked. Looking at day 10, we're just looking at our proportions, and here's our one line with our treatments of Victa, Cleriva, untreated Williams 82, and Jack. What we see at the J2 stage is we have more nematodes with the Cleriva treatment and the Jack treatment. And at the J3 stage, we have less nematodes at the J3 stage for Cleriva and Jack. So we're seeing slower development, at least for those two treatments, at day 10. Let's look at day 20. Again, our proportions and our one line. At the J3 stage, we see more nematodes for Avicta, Cleriva, and Jack. And at the adult female stage, we see less nematodes for Avicta, Cleriva, and Jack. So at least by day 20, we're seeing a little bit slower development for those seed treatments and our resistant variety, and not as many nematodes reaching that full adult stage. Let's look at day 30. We see larger, or much higher amounts of nematodes, at least for the Jack treatment at J3 and J4, and less nematodes at the adult stage. So Jack's working, our resistant variety is working. It's slowing down development. But unfortunately, the seed treatments at day 30, not much different from our untreated control. All right, lastly, once the nematode's in the roots, it's developing, they get fat, the females go exposed, the males go find them. Our question is, do seed treatments reduce the number of females that are able to reach maturity? And does it have an effect on their ability to produce eggs? Our method that we use here is we grow soybeans for either 30 or 60 days in naturally infested soil. Once we do that, we pull the nematodes out of the cone, remove the soil, and spray females off of the roots. And that's what the female would look like there. There. Really hard to see. Spray them off with a high pressure stream of water, and you can count them. Once you count the females, then you can crush them open with just a rubber stopper on top of some mesh and release the eggs. You can count the eggs. Figure out the number of eggs per female. This method is great to show if something works or doesn't work, but it doesn't necessarily show how it worked because there's all those different life stages that we had to cover beforehand. So looking at the 30-day reproduction, first we're going to look at females per gram of root weight. We have two varieties, Jack and Williams 82, and three treatments, Avicta, Cleriva, and untreated. We see differences between our varieties, the resistant and susceptible variety, but no differences across treatments. That's seen again for eggs per gram of root, and again, eggs per female per gram of root. So we saw some differences down the way, but once we get to that 30 or 60-day reproduction, similar to what we saw with that 30-day development, not many differences. So no differences among the treatments, and we saw similar results for 30-day reproduction. And one of the things is these assays tried to be similar to what would be in the field. So they were grown in naturally infested soil. So that means juveniles can hatch and they can enter into the plant throughout the time. In industry, they grow the seed in naturally infested soil for a given amount of days, pull it out, and then grow it, say in a paper towel or sterile soil. So they really are showing, hey, look how effective our seed treatment is. It killed nematodes in the first 5 days, but it doesn't show anything past that. This is showing past that, or that it didn't go past that. So kind of to summarize everything up, the Victa soil leachate reduced nematode hatch. We don't expect to see Cleriva to reduce nematode hatch because Cleriva doesn't affect eggs. It only affects the nematodes within the roots. Victa and Cleriva leachate reduced juvenile speed and change in curvature. Victa and Cleriva seed treatments decreased juvenile penetration at 2, 5, sometimes 7 centimeters, especially with Cleriva. And then our Victa and Cleriva seed treatments reduced the number of females by day 20, but by day 30 we saw no differences. There are many factors that affect the efficacy of these seed treatments, and we tried to explain some of those differences throughout our variety of lab and greenhouse tests. And with that, I'd like to acknowledge my committee members, Greg, Thomas, Gary, Joel, and Santosh. Tilka lab members, especially Augie Beeman, who was kind of my partner in crime for the last 3 years. And the Pandy lab members who have helped me kind of troubleshoot all of this high-tech stuff that's way above my head. And funding is provided by Camtech and Syngenta. And with that, I'll take questions and remind you that it's October 3rd.
So I have a question.
Yes.
Officially, what do the companies label their seed treatments for as far as their time intervals? So you did experiments up to 30 days. Is it supposed to be effective for 30 days, or is it a 21-day window, or is it a season-long window? What is that theoretically?
I have not read anything of a given date, but that doesn't mean that it doesn't exist. I just apparently am not good at reading. I just read a lot of early season protection is what I hear a lot.
So if you're not seeing much efficacy at that 30 days, what's your recommendation then? Is it still to put a seed treatment on to control nematodes?
So if you have a bad nematode problem, like a really bad nematode problem, your resistant variety isn't going to do much, or it's not doing much as it could. So you need to add something else in there. So perhaps in the field we see this under strong nematode pressure, but if you don't have much nematode pressure, perhaps not. I kind of have a prepared slide for that question. Thanks for the plant. So this is just kind of field data of looking at Cleriva complete beans, which is Cleriva with Cruiser Max and Vibrance, versus the orange bar, which is just Cruiser Max and Vibrance minus Cleriva. And we're looking at the reproductive factor, which is just end of season egg numbers divided by beginning of season egg numbers. We don't really see a difference in the reproductive factor between beans with Cleriva or beans without Cleriva.
That's yield. The bars are the reproductive.
Ha ha ha. We don't really see differences between the reproductive factor across those times, and yields are about the same. So take the gamble if you want, I guess.
And you have some data like that for Victa?
Have you guys? I don't have any. I have not. They have data for root knot nematode and reniform nematode down south, especially on cotton, but I haven't seen it on soybean.
We have about half as much data. This is 27 site years of field experiments. We have about 10 site years, and we saw even less differences in yield and nematode control with Avicta. And by less, I mean less than 1 yield and less than 1 reproductive factor.
That's why they don't sell it in the Midwest anymore. perhaps.
They still sell it in the southern United States for control of other nematodes, root knot nematode and reniform, but Clariva was a replacement for Avicta up in the Midwest because soybean cyst nematode is the main one we worry about up here.
So that extra cost for the seed treatment for 20 days of control was pretty questionable based on this data.
So if I can elaborate on Jared's answer for protection, he was right that Avicta and Alivo, a product from Bayer, are described as providing early season protection. Clariva, I've heard them actually say 60 days of protection. And then has anybody seen TV commercials for Nemastrike? I see them every morning 3 times between 6 and 7 a.m. How many days of protection do they say? Do you all recall? They say 75 days of protection. I don't know how they come up with those numbers, but to kind of round out the answer to your question, they're now starting to give specific numbers of days, although I don't know, I've not seen the data to back that up, but they are giving numbers of days.
Yeah.
Does the cyst protect against pathogenic fungi?
There are microbes that are able to enter into the cyst, and we've seen them there before, but I would assume, or I would not assume, my thought is that the cyst is there to protect the eggs rather than being outside barren in the soil, so that's why they're able to last for so long. Yeah.
So, at the beginning you talked about how the roots themselves release either some chemicals or nutrients which induce the eggs to hatch. So presumably people have tried to look at what those exact chemicals or nutrients are. Have people tried to put those in seed treatments for a non-host crop like corn? So you could grow in an off year, grow that, and then try to induce nematodes to release and then decrease the population?
So you looked at glycinoclepin A, which is from kidney bean, and that's a hatch stimulant, but it's very expensive to produce it, to put on a treatment for a non-host crop, I feel like.
We looked at it in the context of a soil application. I worked with a chemist in the 90s. We tried to make it synthetically. And it was really expensive, as Jared said, to make, and you would need gallons of it for a field. But that was back in the day before anybody dreamt of seed treatments for nematode control. So I don't know if it would be more economical or practical as a seed treatment.
I'll
I think there's
suggest
no
that
great
during
trap
interviews.
crops that you can use. At least for this particular, for soybean.
Well, I mean, they're defined great.
Something that would also be economical to use.
So corn, a lot of the juveniles, why don't you talk, the juveniles going into corn roots? I mean,
So the nematodes don't differentiate between what plant they're going into. They're just gonna go into a plant root. They're gonna try and find the inner part of that root and start feeding. It's that interaction between the proteins that the nematode secretes into its feeding site that help it develop that syncytium for that very specific interaction with the soybean plant. So there are a few bean plants that they can produce small amounts of syncytium, but for the most part, corn, it's an incompatible host, it won't produce that. So if you can find a host that it can't produce the syncytium on.
yeah, so the worms, once they hatch, they're relatively dumb. They'll go into corn roots or soybean, anything that's a root, and then they get in there and they find out it's not a host. So as Jared pointed out, kind of the weak link in the life cycle, they're vulnerable when they're hatched until they find a compatible host root to get into. So corn is a trap crop, and we can see, what do we say, as much as how much of a decrease in 1 year of corn?
Much as 50% decrease in egg density in the soil after a corn year.
But it seems to be tempered by the amount of hatching that's occurring, not the amount of infection that's occurring. Other questions? If not, there's one more.
One last one, you almost made the cut.
Is this treatment system
Systemic? No, so Avicta or Clariva will only work on, it only attaches to the juvenile cuticle. So that's not going to move through the plant at all. And then Avicta, not to my knowledge that it's systemic, it just stays where it was added, just along the root. We'll see how far it moves though.
So it can move on the surface of the root, downward. We don't know how deep. But. Okay, if there are no other questions, let's thank Jared one more time.